type menti.com and then enter this code. One 97029742. So when you do that, you will be faced with a question to order the steps that you would use for next generation sequencing or high throughput sequencing. I don't know if you guys can see any of that in your can you guys see the screen, the PowerPoint?
Yes.
Okay, so the question is, what are the steps for next or high throughput sequencing? Maybe I should just put it here. What are the steps for high throughput sequencing? This is on Menti. You have the code here, which is 17029742. And I think the Menti platform is now running for you to answer that question. And I can see several respondents have already done that, so thank you. Please order them according to, and this is a team thing, so whichever is ordered first by more people will be number one. Whatever is ordered by the fewest people as number last will be last. So just arrange them. If you think sequencing is number one, if you think bioinformatics is number one, do that. So far we have 22 respondents. I need at least 100 because I can see you guys are 170 now. So please participate. Type the code, type menti.com onto your phone and then enter the code 17029742, I think. I'm looking at Menti now, not the screen, not the PowerPoint. We are at 30. I need more, guys. I can see some changes. Please vote for your favorite. I can see more changes. Good. Now that we are at 60, I can share the Menti screen as you continue to answer so that you can see how you are responding. So keep going. I hope you can still remember the code. Menti.com, the code is 17029746. I'll type that in the chat. Menti.com. Code is 17029742. I hope I got that right. So we are at about 82. I'll stop this at 100, so please vote. Whatever is on the screen is not necessarily correct, so please vote what you think is correct. Don't be swayed by the masses. Okay, we are at 100, so I can talk now. So what you see here is actually an ordered way of the sequence. I think extraction, we all agree, comes first. Before library preparation, we do an amplification. But I can see why we would think that amplification comes after library prep, because there's more amplification during sequencing. So sequencing, as remember, cluster generation, which includes amplification. But before you do library prep, you still need to do some amplification. So, but I can see bioinformatics comes last, DNA sequencing comes second last. These two are the ones that we can debate about. So I would think, yeah, I wouldn't mark you wrong for this if you said, but I was expecting you to bring DNA amplification first, because it's you amplify before you can make a library, because the library is being made from a high volume or high concentration of DNA. But thank you very much. I can see you retained quite a bit. So we'll move on to today's topic, which is bacteriophages, or phages in short. And phages are really viruses, but they are not viruses that infect humans. So any reason why we are studying, anybody, any guess why we are studying viruses that don't infect humans? Anybody, please post in the chat. Why do you think we are studying phages, these viruses that do not infect humans? They infect bacteria. Okay, I can see some responses. Let's see if you guys are right. The outline for today and what you guys would expect you to understand or be able to do is to describe what is a bacteriophage, what is the general structure of a phage, what is the classification. We'll make this very simple because I mean, it can be quite complex. They're very diverse. We'll also, and this is probably one of the important things to understand, the difference between lytic and lysogenic cycles. We'll explain that. We'll also discuss applications or where phages are used or where they're important, and give some special emphasis to one of these applications, which is phage therapy, its challenges and its advantages. I would like to say at the outset that right now, I can see somebody asking what is the topic, so I'll flash that again. I don't know whether you guys can see my screen or not, because I'm seeing some questions that are suggesting otherwise. But the first thing I would like to say on the outset is phages are not yet licensed for clinical use. So do not live here and go prescribing phages for everybody in the wards. We will see why that is not the case, but there are places where they are used for clinical applications, especially in Eastern Europe, in Georgia, especially which was part of USSR. And at the time, USSR had been cut from the rest of the world. And so they could not get antibiotics readily. They perfected the art of using phages. Okay, so we proceed. What are phages? A phage is a virus that attacks a bacteria. The good thing is that most phages will attack a specific bacteria, but some will also affect or attack several different bacteria. The same way some viruses are very specific to one host, and some that have a broad host range. The other thing about phages is that they are ubiquitous. They are everywhere. They are in our bodies, they are in sewage, they are in seawater, they are in soil. Anywhere you can find phages, and this is because anywhere you can find bacteria, you will find phages. And to one bacteria, there will be hundreds of phages because they feed on bacteria. Some of the magical healing powers attributed to water bodies across the world was actually due to phages. For example, somebody isolated phages that could cure cholera in one of the largest rivers in India. And we know there are other phages that can also treat wounds because of they have the effect of treating or killing the bacteria in those wounds, be they pseudomonas or staph aureus. And so I'm sure you've heard in many legendary stories, be they in biblical or other religious books or in folklore, that certain waters have been associated with healing powers. This is one of the things that has been scientifically proven, that phages play part of that mystery. So selectively, phages are known for selectively attacking specific bacteria, and they spare other bacteria. So if, for example, you gave somebody today antibiotics for osteomyelitis, and they are taking this for 6 weeks, for instance, what happens in the gut is that you clear most of the normal bacteria, the normal flora that you need. Because remember, your gut, especially the colon, is full of bacteria that serve positive roles, from metabolism to immune regulation and what have you. And when you clear the normal bacteria, the abnormal one, like Clostridium, for example, which you will learn about, can cause diarrhea. It is causing diarrhea because the normal flora has been cleared, there's no competition. So some pathogenic bacteria which are resistant to these antibiotics that you're giving will cause problems. Another common illustration is what we see with especially ladies when they're taking antibiotics for a while, and they end up getting vaginal thrush or candidiasis because the bacteria has been cleared and now candida overgrows. So candida is there as a normal flora, but it's controlled by the other bacteria. So having normal flora in our bodies is a very good thing. And right now, as you will learn about bacteria and other pathogens and other, should I say, organisms, you will learn that microbiome is playing a very big role in our immunity, autoimmune disease mediation, cancer, and what have you. And we are actually just beginning to scratch the surface. And so when you have therapy that is narrow spectrum, for example, if you're treating a patient, you choose the most narrow spectrum antibiotic to save other organisms, and also to reduce the risk for antibiotic resistance. So it's a good thing. Don't always look for the big gun. You know, you have a sore throat, you want to go for the strongest, as they call it, antibiotic. No, look for the most narrow in spectrum to treat your organism. The only problem is when you don't know what you're treating, then we tend to go for empiric therapy, which is broader spectrum. But if you have a lab report telling you this patient has streptococcus, you know streptococcus responds very well to amoxicillin, as you will learn, then you give them amoxicillin. So it is good that bacteriophages only attack specific bacteria and they will spare others, so they preserve normal flora. They are used in phage therapy, and this actually was developed before antibiotics were really developed, but because of challenges with the development, and because antibiotics were much easier to work with and to prescribe, they overshadowed phages. But now they are taking a lot of interest because of antibiotic resistance or antimicrobial resistance. As we get bugs that are resistant to most of the antibiotics we have taken more interest in phage therapy. And actually they have been licensed in food industry to prevent colonization of certain products like poultry and beef. Okay. So this again, please don't grab these numbers, just to show you the relative size of bacteriophages, so that you can see how they'll fit in a bacterium. And usually we have viruses around the same size as phages, although they're on the lower side of the virus scale. But bacteria will be several times, maybe 10 to 100 times larger than phages. And so you'll find that a bacterium can hold hundreds of phages. So when they multiply in the bacterium, there will be hundreds of them being released, or at least tens, depending on the size. So that's how they're able to replicate within the bacteria. So because they're much smaller than the bacteria. That is the main point of this slide. This is the structure of the prototype bacteriophage. There are other different shapes, as we'll come to see, but this is the prototype. And it has, if you start from the top, a head, which looks like, which is a capsid. It's icosahedral. You can see the different shapes. And this contains the genome of the phage. Just like other viruses, these phages can carry DNA or RNA. They don't carry both. They can carry DNA or RNA. And the DNA can be double-stranded or single-stranded. So quite similar with the virus, with the virology we know so far. And the head is attached to a tail, and the tail, as you can see here, has several structures. It has the spikes for injecting the genome into the bacteria, and the tail fibers for attaching to the bacteria. But generally, you have a head and a tail, and for some of them, the tail is contractile, but for others, it is rigid. It doesn't contract. And you will see, usually when this contracts, basically you're bringing the collar closer to the base plate, and then that injects the genome into the bacteria. So this is really the shape of the structure of a bacteriophage. There are a few other things here that we don't bother with too much. So you just need to know that the head has this genome, and then the tail has the structures for attaching and injecting the genome into the bacteria. This is an electron microscope, micrograph of the of the phages, and you can just see the tail and the head. For some of them, the head is oblong. For others, it has symmetry across all axes, like you can see on your right. This one is oblong, it will probably just have symmetry on 2 axes. This one has more or less, all the sides are equal. This one is a bit oblong. So these are the different structures you will see, and additional ones as you will see shortly. So I'd like to show you a video right now so that we can probably see some of the
Take a look around. From the big to the small, the visible to the invisible. Life is everywhere. The smallest unit of life is called the cell, and your body is made up of 30 to 40 trillion of them. But if you took the total number of bacteria on your body right now, that number would outnumber your body cells 10 to 1, making us theoretically 90% bacteria. Now that's a lot of bacteria, but can you imagine the total number of bacteria on Earth? It's a ridiculously high number, and we all know bacteria can survive almost anywhere on the planet. Now take that total number of bacteria on the planet and add to it every other living thing, from birds to mammals, fish, add it all on. What number do you get now? What's the total number of living organisms on Earth? What if I told you that it didn't matter because there's an entity on Earth that outnumbers every living thing combined that you guys probably don't even know about? Bacteriophages.
Bacteriophages, or phages for short, are viruses that infect bacteria cells. They are by far the most abundant and diverse biological entities on the planet, totaling 10 to the 31st in population. To put that in perspective, there are 1 billion times more bacteriophages on Earth than stars in the observable universe. There are 10 billion times more phages than every grain of sand added up on Earth. Lastly, if you added the net worth of Bill Gates, Mark Zuckerberg, and Warren Buffett together and multiplied that number by 1 trillion, you'd still have 100 million times less money than the total number of bacteriophages on the planet. So should we be worried with all these viruses floating around? Well, no. These viruses are very host specific, meaning they only infect certain species of bacteria cells by recognizing very specific receptors on the surface of bacteria. In other words, they're no harm to human cells at all. Bacteriophages were first co-discovered in the early 1900s by a British scientist named Frederick Twort and a French-Canadian scientist named Felix Durel. These scientists noticed that clear circles called plaques were forming on their petri dishes that were growing bacteria, suggesting that the bacteria was being killed. Durel named them bacteriophages, literally meaning bacteria eaters. These phages solely rely on random encounters with bacteria that they infect. Once they come in contact with their specific host, they attach to the surface of the cell with tail proteins and begin to degrade the cell wall. They then inject their DNA or genetic material, which is stored in the head or the capsid, through their tail and into the bacteria cell. Depending on the virus, the cell does one of two things at this point. If the phage is a lytic virus, the cell's machinery, including the ribosomes, will begin to make viral proteins that were coded in the newly integrated viral DNA within minutes. The cell continues to replicate the phage progeny until eventually the cell bursts open and hundreds of newly created bacteriophages are released into the surroundings, looking to infect more bacteria. If the phage is lysogenic, on the other hand, it does not burst the cell open immediately. Instead, the genetic information is replicated along with the rest of the cell's genome and remains dormant. Once the conditions are right, that's when the viral replication initiates and lysis of the cell occurs. So what's the big deal? Why do we care about bacteriophages if they don't really affect us? Well, first and foremost, the potential antibiotic capabilities of these phages are astounding. In a world where antibiotic resistance is on the rise and superbugs are always a threat on the horizon, phages could offer a quick and inexpensive way to solve these bacteria problems. Treating bacterial diseases like tuberculosis and leprosy could potentially be achieved by introducing phages that infect that specific bacterial strain. Phages also have some bioremediation potential and could be used to control environmental problems in the future. Hospitals have been attempting to develop ways to use phages to reduce biofilm in hospital settings, which is becoming an increasing problem. Lastly, the US Food and Drug Administration and the USDA have approved several phage products, including treating meat and poultry products with phages. A ton of research and studies continue to take place on bacteriophages, many of which involve their genomics. Because of their abundance, there is a humongous reservoir of untapped and unsequenced gene information that could lead to the next big discovery in science. Who knows what's to come? Thanks for watching.
Okay, so that is just to introduce phages to you. We will discuss the lysogenic and lytic cycles further. I know they were not adequately covered in that video, so don't worry about it. I'm now going to share the PowerPoint again. Yes, I hope you can see it now. We are now looking at classification of phages, and there are different ways of doing it. You may see the classification by genome, which is the DNA or RNA. And for both of these, you can have double-stranded or single-stranded DNA or RNA. You will also see classification by the shape. The typical bacteriophage will have a head and a tail. Some will only be head only, and some will only have the tail. So all these are possibilities. Some are even irregularly shaped, but they will generally fall into these categories. The other one, which is more considered more scientific by ICTV, is classification by host, so that a virus or a phage that infects E. coli will be called Escherichia virus. A phage that infects Salmonella, this is the bacteria that causes typhoid, will be called Salmonella virus, and several others. Basically, almost every bacteria will have its own or several of them. And this is naming by host. There is also the cycle behavior, and this is very functional because we consider the application of these phages depending on how they behave in the body or in the host. And this can be lytic, which is basically a phage that will rupture the bacteria once it enters it. And it does so by replicating continuously. So lytic phages are the ones that will get into their host and immediately begin to replicate. And by doing so, they degrade the bacteria and eventually burst it and kill it. The lysogenic ones will not begin replicating immediately they enter their host. They first integrate their genome into the host genome. And as the bacteria copies itself, or as the bacteria makes copies of itself, it will also be copying the temperate phage or the lysogenic phage. And eventually, when the conditions change and the bacteria is exposed to tough conditions, say drugs, or in the lab we use UV light, it then becomes lytic and begins to replicate. These are just the shapes that we have. Again, I don't expect you to cram this, it's just for you to see what you have. You can see the first and the second are almost identical, except that is A and B, except for the tail. A has a contractile tail and B has a rigid tail. C has a shorter tail. D has no tail but has large bodies, as you can see at the corners of the capsid. E, the only difference with E and D is these bodies that you can see here. and F is headless, or it's just a tail. So you can see the different variations. And in fact, this is a very simplified approach. There are bacteriophages that will have very interesting shapes, although generally they'll fit into this. So when you see a bacteriophage looking like a slug, don't worry about why. You try and fit it into these categories. You can also classify by genome, and this you can see is double DNA and RNA to the right, DNA to the left. For the DNA, you can have double-stranded or single-stranded. And within the double-stranded, you can see the different shapes that come. These are basically A, B, and C. And for the single-stranded DNA, you have this one with the large bodies on its capsid, and the one that has a tail only. And then RNA, you have, again, tailless virions. Some even have what looks like an envelope around them. So quite interesting shapes. Again, I will not necessarily ask you to classify phages. This is here just to inform you of how they look like. I'm more interested in the applications because that's where your usefulness will come in. The phage cycles, and this is important to understand, it's good to distinguish between the two cycles. I've said it before for emphasis, so I'm going now to describe it in a little more detail. But in short, a lytic phage will replicate immediately it enters the host and kill the host. A lysogenic phage, when it enters the host, will first of all integrate and remain quiescent or latent, so to speak, until the conditions change. And usually in the lab, we change these conditions using UV radiation so that the bacteriophage is triggered to move from the lysogenic cycle to the lytic cycle where it replicates. So if we are to start at the top, what you have as this oblong structure is a bacterium. The bacterium has the genome, the DNA material, as you can see, in circular form. And when the E. coli, say the phage, infects the bacteria, you have two options. On the left, the bacteria, if the phage is lytic, and some of them can swing from lytic to lysogenic cycle. Some of them are more lytic, others are more likely to be lysogenic, but generally, most phages will be able to swing between the two. So when the E. coli enters, the phage enters the bacteria, what enters is actually the genome of the phage. It's injected into the bacterium, and it will quickly begin to be translated into proteins. And these proteins will degrade the genome of the bacteria, break it into pieces basically, and will then be assembled into the new phages. And as the number increases, this bacterium will burst. It can no longer contain the hundreds of phages inside, and then they will be released to go and infect other bacteria. The option 2 is when the phage enters the bacterium, but does not initiate replication of its genome and synthesis of its proteins immediately. It will inject, or it will integrate its genome into the bacterial genome. So you now have a circularized bacterial genome, which now contains the phage, as you can see this zigzag stretch here. And it will stay like that as it will not cause any harm to the bacteria, but as the bacteria copies itself, bacteria, you will be told how bacteria reproduce, but they make copies of themselves through binary fission. They will also copy the genome, and the genome that they're copying will contain the phage. And this can continue, and the phage will be passed through many daughter bacterium cells until something happens. In the body, it could be introduction of drugs, which can put pressure on the bacterium, or it could be in the lab, because this is a process we use in the lab to introduce new genes to a bacteria for one reason or the other. You can use phages to introduce a gene into a bacteria. And when you want the gene to be expressed, then you induce the lytic cycle by UV irradiation, basically exposing the bacterium to UV light. And so the phages go into the lytic phase. They begin to replicate within the bacteria. They kill the bacteria or lyse it, and then they are released to go and infect new phages, new bacteria. So I hope that is clear. If it's not, there's a link to a video here. You can also check and see if it helps. But in short, lytic phase kills the bacteria. Lysogenic phase will not kill the bacteria. This is the cycle that lytic phase follows in a little more detail again. Attachment is through the tail fibers with the spike injecting the blue genome here is from the phage. You can see the tail has contracted to inject it. And then the bacterial DNA is sitting pretty here, but soon as the bacteriophage begins to transcribe and generate several bacteriophage proteins, they degrade the genome of the bacteria and use the bacteria system to make copies or to make or synthesize proteins that are required for the phage. And then eventually the phage will be assembled, several of them in one bacterium. Eventually that will continue until the bacterium is lysed and the phages are released. And you can see they are being released with the copies of the genome inside, just like the mother phage. So this is really the long and short of the lytic cycle. Now, we have, dependent on these cycles, transduction. And you will learn later in bacteriology that transduction is one of the ways through which bacteria transmit resistance to each other or transmit genetic components to each other. We also use this transduction in the lab in molecular biology to generate certain proteins. And usually bacteria are very good for generating proteins. So for example, if you wanted to generate a certain antigen and it has a short code that you can insert in a phage, you can use that phage to infect bacteria, and some of the bacteria that survive may express that antigen. So you just let them multiply as many times as you can, then purify it. And usually you know whether it has worked by including within the new gene that you're inserting a gene for resistance. And maybe so that if the phage succeeds in inserting the genome into the bacterium, there is some resistance to, say, amoxicillin, and then you can treat the entire culture with amoxicillin so that the bacteria that survive, you know for sure they have the gene of interest. The ones that die will not have the gene of interest. So that's just an illustration of one of the ways you can apply this knowledge in the lab. But the transduction process, as you can see here, you have in stage A, infection of the bacterium by a phage going on, injection of the genome, and then in the second step, the phage will, this is a lytic phage, so it will begin to replicate, degrading the bacterial DNA. And then during assembly, what you see now during assembly is that most phages will just pick their normal genome, but a few phages will pick part of the bacterial genome randomly. So this bacterial genome picked randomly could be, for example, a cholera toxin or any other bacterial toxin, or a gene that codes for resistance. And so what you're seeing happening randomly, because these are millions of daughter phages also multiplying in millions of bacteria, what you see then is just an element of chance. It's not planned. And if these few phages that have now picked a genome or a gene from the bacteria infect new bacteria, they will transfer this gene to the new bacteria. And because now they do not code, this new gene does not code for phage proteins, it will not be able to kill the bacteria. So what happens is that the gene or the genetic material that has been introduced will now be available to code for whatever it was coding for. So if it was a toxin or a resistance gene, then it will now be available for this bacteria to code for that product that it is meant to code for. And if it is beneficial to the bacteria and the bacteria survives, it will be able to pass it to daughter cells so that the new bacteria that are being produced, if it was a resistance gene, are now resistant to whatever drug that was there, or they're able to produce a toxin that can harm the host. So this is again by chance, and it only happens, we only see it when it benefits the bacteria, so that the new bacteria has been able to pass genetic material. The old bacteria that was dying has been able to pass genetic material to the new one that has been infected by the defective phage, and then it is now able to create a new clone of bacteria that has that genome or gene that gives it some benefit. So this is one of the fears we have actually with phages, that it may be possible for them to transmit or transduce harmful genes to new bacteria. But usually if the phage is lytic, it will not be a big concern. Usually it's a bigger concern with the lysogenic phages, as you can see in this one. And this is the one that we actually use more in molecular labs. This is again the bacterium which is being infected with a phage, but this phage is not lytic, it is lysogenic. And so the genome of the phage will be inserted into the bacterial genome, and we are targeting this gene called GAL. This is a gene for metabolism. I won't go too much into it, but somebody is interested in this gene. So what you do, once the phage enters, it will be randomly integrated into different parts. But as the bacteria multiply, if you induce a lytic cycle using UV radiation, this will be broken, or the genome of the bacteria will be broken into various components, and by chance, some of the phages will pick up the gene of interest, for example, this gene here, and if they pick it up, then the normal ones that have only the phage DNA will continue to infect other bacteria, but the one that has picked up, say, this gene of interest will also have a component of its own phage DNA. It may be able to infect other bacteria, but it will not kill them so much. It will not, the bacteria will now get this new gene that you're interested in, but they will not be dying. They will now remain in the lysogenic cycle. And so you can express this protein for whatever purpose that you want it to. So transduction is one of those procedures that happens naturally, and it is mediated by phages in the body, but we've taken advantage of it also in the lab to express certain proteins. And how you know which bacteria have the gene of interest or not is by including, as I said, a gene that codes for resistance, so that when you treat the culture broth with that drug, or that you know that the bacteria that will survive have resistance against that drug and also have the gene of interest. So when you culture that bacteria, you'll get a lot of the protein that you're interested in. So this is transduction. It is one of the things, again, we learn in bacteriology, how bacteria transmit the resistance genes and other genes across each other by the assistance of phages. Now, applications. There are many applications. One is phage therapy, which we've mentioned several times today, but you can also have phage typing. Now, what is phage typing? Imagine you have several types of bacteria. Somebody may be, whether you're doing research or you're doing some sort of even diagnosis, although it's not a standard form of diagnosis, and you have bacteria that say you have A, B, and C, bacteria A, B, and C, and you have a phage that you know only attacks A, but you don't know which of these bacteria A, B, and C is actually identified as A, say Salmonella. So if you culture all these bacteria together with phages, the one that the phage attacks is the one that you know is the host, because you already have characterized the phage, and you know the phage attacks this specific bacteria and not the others. And it could even be strains of bacteria that you're targeting. So phage typing is using phages to identify bacteria. Phage display is what I've briefly touched on, using phages to synthesize proteins, which you can then study. And a lot of this has actually been replaced by plasmids, which is another thing altogether, but it still happens. And gene delivery, if you want to deliver certain genes, you can also do that using phages. Vaccine delivery has been tried for phages, although we don't have any human vaccine that uses phages. And also biocontrol, which is control of plant diseases using phages. And you saw it in food as well, in preserving food preservation of especially poultry and beef, which is one of the applications that has actually been licensed for. So what is phage therapy? This is a form of therapy that was developed about 100 years ago, but fell out of favor when antibiotics were discovered because they were easier to apply, and even one antibiotic can cure so many other illnesses. So for example, if you gave somebody ceftriaxone, you would be able to use it for meningitis, for pneumonia, for sepsis, for different infections. Although right now we know ceftriaxone resistance in many places is around 50%. So we are no longer using it so much. We have overused it. But that was the beauty with antibiotics. Unfortunately for phages, you have to get a new phage for every bacteria. So if you meet Staphylococcus in a wound, you need to get a phage for Staphylococcus, and maybe now you can keep that phage for the next Staphylococcus. But if somebody else comes with Streptococcus or another bacteria that is not Staphylococcus, then they will need a different phage. They are currently being explored for treatment for drug resistance and even chronic infections. And we usually use lytic phages because they're the ones that will kill bacteria with a lower risk of transmitting transduction, as we've seen before. So we prefer lytic phages because they'll kill the bacteria before they get a chance to transduce. The good thing is also the autodosing. Autodosing meaning, because this phage is targeting a specific host, when the host dies completely, then the phage will also die, because that is it relies on that phage on that host to multiply. So if it can't multiply, then that's it. So autodosing is important in this sense. It also has great potential, but we still need a lot of data on safety and efficacy to achieve general acceptance and also licensure. The concerns about phage therapy, this is why you cannot go tomorrow to Ward 6 and prescribe phages to a guy with osteomyelitis. It is because, one, you need to find the phage which is effective for that bacteria. It means you need to pick that patient with osteomyelitis or with a chronic wound. You pick a sample in the correct manner. A lot of us just swab the pus, that is wrong. You go to the base of the wound, clean it up, the wound kabisa, go to the base of the wound, collect some tissue, and then take that to the lab, they culture and find out which bacteria is there. If they find it's Pseudomonas, for example, you then find a phage that works against Pseudomonas. Fortunately, phages are all over. You can even go to the sewage or the taps in KNH, and you will find phages there. And probably they'll be targeting the bacteria that is circulating there. So, but you see that takes time. It's not like going to the chemist and buying some drugs. And then you have to grow the phage, quantify them, and then now apply. Eventually, you can prepare certain preparations, and now there are preparations that are bottled for specific conditions like wounds. And you know this preparation has 10 phages, one for staph, one for this bacteria, another one for this other bacteria. So then in that case, they are already prepared. But generally, you need to identify the bacteria first before you can get the phage, a phage that works for that bacteria. So that slows things down. But if somebody has a chronic wound that is not healing, then there's sufficient time for it. The other concern is that some phages may transfer genes that are code for bacterial toxins or antibiotic resistance. And that is a concern we have a lot, especially for lysogenic phages that can allow bacteria to multiply with these kind of toxins or genes. And we also don't fully understand the pharmacokinetics and pharmacodynamics. How does, I hope you guys are doing pharmacology, but how much do you need of this phage? If, for example, you want to treat a chronic valve infection in the heart, how much of it will get there? How much of it will go elsewhere? And what are the effects of that? So there's still a lot of that we do not know. There has been a concern, although I don't think this has been observed, that phages may trigger a humoral immune response This is actually true, actually. The next one, the anaphylactic shock, is one that hasn't been observed. But when you introduce somebody to a phage, or introduce phages to the body, the phage are attacking a bacteria, but the body will also detect them as a foreign organism. And for that reason, they will generate some neutralizing antibodies to that phage. And next time you try to apply the phage, it may not work. Interestingly, also bacteria have the immune system. And if you are curious, you can look at CRISPR-Cas9. CRISPR-Cas9 is the immune system of bacteria. And when they are infected with especially lysogenic phages, but also other phages, they can detect that and call and get resistance by coding for immunity against that bacteria using Cas9. Cas9 is basically a molecular seizure that when it encounters a phage, it will store a special sequence of that phage in its own cartridge in its own system, so that next time it encounters a similar genetic sequence, it will excise it and so render that phage non-functional. So we have humoral immune response from our own bodies against phages, but bacteria can also develop resistance against phages using their own immune system, so to speak. The risk of anaphylaxis is usually due to bacterial endotoxins from impure products. And there is also fear that if you lyse bacteria too rapidly, they may release proteins that can also cause toxicity, sepsis. And what we are talking about here is that for you to produce phages, you need bacteria. So you can imagine you have a culture broth of, say, E. coli, which you are using to produce a phage that can actually treat E. coli in the body. So as you do that, these phages are multiplying within the bacteria. So eventually you have a broth that has both phages and bacterial proteins. How do you purify that broth to get only the phages without carrying any bacterial endotoxins? Because if you introduce the bacterial proteins into the body, they may cause some form of anaphylaxis, especially if it's through injection. So that is the basis of this first part of the last sentence. And also the fear that if you introduce phages into the body, they may kill the bacteria so fast that you get a lot of bacterial protein released, and that can also cause problems. Fortunately, this has not been observed, mainly because most of the phages have been used for localized treatment or through local application. So it has not been observed. It's more of a concern from safety point of view, which still has to be addressed. Now, so what is the difference between bacterial phages and antibacterial agents? The advantages for phages that they are specific to the bacteria, while the antibiotics may have a more broader reach, and so they kill even normal flora. They occur abundantly in nature, so they are easy to get relatively, and they have a good safety profile so far. You will not hear about kidney failure or liver toxicity with phages because they do not undergo that metabolism like antibiotics. There's also low selective pressure on bacteria in the sense that when you apply drugs, they apply pressure on the bacteria, and that selects for bacteria that is sensitive, that is resistant, and it will let that bacteria that is resistant to the drug thrive because it has killed the others. The phages will generally be able to attack all the bacteria without, the bacteria target bacteria with E. coli without selecting for those, unless now they develop resistance. Harmless to humans because they don't attack human cells, and they are relatively easy to produce because all you need is a culture of bacteria and then throw in your phage and it will be produced. The challenges are that bacterial resistance can also develop against phages. As I indicated here, you can look at the CRISPR mechanism, and immunogenicity against phages by our own bodies can produce anti-phage antibodies, which will then make, if for example, you need a second application a week later, it may not work. The narrow host range is also a limitation. So if you have a patient with a wound and you can see the wound is infected, but you don't know which bacteria it is, it is difficult to apply just one phage because you don't know if it is targeting. You may know the common causes, but usually it's 1, 2, or 3 bacteria that you need to target. And that's why we have antibiotics that say this is for gram-negative organisms. This is for gram-positive organisms. And by using that kind of range for antibiotics, we're able to give empiric treatment. But for phages, we cannot give empiric treatment. We first have to test and confirm what is causing that condition. No regulatory approvals for routine use. That means you can't walk into KNH unless you are working under a research protocol and prescribe phages just like that. And also co-evolutionary dynamics with bacteria, because these are 2 organisms. As they evolve, we don't know what they portend. And so this is an area of curiosity and concern, because when you expose bacteria to phages, they will try to evolve through selective pressure again to beat that phage. And then the phage may also evolve to be able to infect the bacteria. So eventually, we don't know what else can happen. And phage delivery to the target site is a challenge. It's easy to apply a phage to a wound or to a surface that you can access. But if it's somewhere in the body and you have to inject or directly deliver it, that could be a challenge. I need to finish this by talking about antimicrobial resistance, because antimicrobial resistance is an important part of virology and also microbiology. We need to appreciate that antimicrobial resistance, and this week actually being the week of World Antibiotic Resistance, it is one of the top 10 global health threats today, resistance to drugs. And it's mainly driven by misuse and overuse of antimicrobials. You are treating a patient, they have a condition that is viral, but they are demanding to have antibiotics or antibacterial agents, or you are using a low dose or using the wrong one, and this can push a lot of issues. We have seen people die from pneumonia because the pneumonia is there, but you cannot treat it because there's no medicine that can work for it. We also have hundreds of thousands of people dying every year because of such resistance. And this is increasing, and it's actually increasing more in our part of the world, sub-Saharan Africa, than other parts. If you go into any chemist today, you will buy any antibiotic that you want over the counter. If you go to the developed world, you need a prescription to buy any antibiotic. There's also a huge financial cost, both to the health system and the economy, because when somebody is infected with a drug-resistant bacteria, they have to stay longer in the hospital. That means they're not working, and somebody probably has to stop working to come and take care of them, which really is a negative thing, considering that work is what generates income. And also, as they stay in the hospital, they're going to have to pay for it. It is unfortunately a complex problem, but it will require a lot of collaboration to work on different aspects, from preventing infections like using vaccines, appropriate antimicrobial use, meaning if somebody has, you suspect somebody has an infection, you find out by doing a culture and sensitivity what is causing it and what it's sensitive to. And even when you start empirical drugs, before you start, you take the sample, take it to the lab, so that by the time you are reviewing the patient, you know better what they have and whether to change the drug to one that is more narrow spectrum or to one that is working. And then diagnostic stewardship is about how we collect our samples and how we make sure that the sample is the right one, it is collected in the right way, and it is tested without a compromise of quality. Monitoring is also important because we need to know how much resistance is in circulation, and we are not doing this in humans alone, because in animal health we know people administer drugs to improve the growth of the chicken and other animals, and even now people everywhere will tell you when animals have this problem, go and buy this antibiotic. We use the same antibiotics in humans. So when you're eating chicken that is already been treated with certain antibiotics, you're being exposed to low levels of those antibiotics, and sometimes that may select for drug resistance. And also the environment being contaminated with antibiotics can select for drug-resistant bacteria, which will then get into our food chain and cause infections. So phage therapy videos are here. I will not get into them. I've just shared them for those who have time to watch. But now I would like to ask one question, and then before you leave, we have an assignment for the afternoon, which all of you must do. So check, wait before you leave. But this question is again on Menti. Which one of the following statements is true? You can go to Menti as we look at this. But one is, we use lysogenic phages to treat drug-resistant bacterial infections. Is that true or not? Phages are usually broad spectrum, targeting several bacterial species. Is that true or not? Phages are licensed for treatment of chronic infections like Brucella. Is that true or not? If applied in high doses, phages can attack human cells, and phages may include production of phage neutralizing, may induce phages, production of phage neutralizing antibodies in humans. So I'm just going to start the poll now in Menti. You can go to Menti now and begin to vote. Just vote for what you think is true. Again, the code is the same code we used before. I can see there are quite a number of comments today in the chat, and we'll take a look at that as well and see. So we are now at 53, 60. Again, once you get to 100, I'll share the screen. So these are your choices. Most people have selected that phages induce production of phage neutralizing antibodies in humans, which is true. Nobody chose the second last one, which says that if applied in high doses, phages can attack human cells, which is of course not true. And a few others have selected the others. Let me see the first one. We use lysogenic phages to treat bacteria. We actually use lytic phages to treat bacteria, not lysogenic. Lytic are the ones that kill bacteria. The second one is phages are usually broad spectrum. As we said before, phages tend to target one specific host. That's why you have a phage called Salmonella virus or Escherichia virus, because they target that particular host. The third one, talking about phages being licensed for treatment, there's no licensure right now, although we know there is research, what we call compassionate use or research-based use, some clinical trials ongoing. So yes, thank you very much. It looks like you've absorbed quite a bit. I'm now going to look at the comments, and then after that, I shall give you an assignment to do in the afternoon before I join you later at 3:30. So I'm going to share my the PowerPoint again. I hope you can see it now. And as we share now I'm just looking at your comments, so maybe I should share my screen instead. But I'm trying to share my screen so that you can see my comments. So my screen is quite a number of comments, and I know I don't need to respond to all of them, so I'll just select a few. Maybe I should start at the bottom. So lysogenic cycle doesn't destroy bacteria too, but you can convert a lysogenic bacteria to a lytic one by inducing tough conditions. A lot of these answers. Low selective pressure. Yes, it doesn't mean they have no selective pressure. They do have, but it's lower than antibiotics. This is true, Kelvis Maranga, that if bacteria become resistant to antibiotics, if you combine that same antibiotic with bacteriophage, you may overcome that resistance. But this has been shown in the lab. We haven't had such applications in the body yet. As bacteria gains resistance to phages, does it affect resistance to antibacterials? Yes, we've seen some lose the genome, the resistance genes, but it's not always the case. Okay. The advantages I was listing are for phages, not antibacterial agents. Using phage therapy, does using phage therapy provide permanent immunity for the next infection? Well, once the bacteria is cleared, the phages will also be cleared. So if the infection recurs, we will need a new dose of phages. You can see somebody mourning the recent letter we saw from KNH. But conjugation must happen for transduction to be complete, not necessarily. Conjugation is a totally different thing. You will learn about that in bacteriology. It's not the same as transduction, and they are not necessarily occurring together. So somebody is asking that I said that the lysogenic cycle does not cause lysis of bacteria. So how will the phage be helpful in killing bacteria? Good question. We only use lytic bacteria to kill phages to kill bacteria, not lysogenic ones. So you can still learn bacteriophages under bacteriology. They are under virology because they are viruses. And also it's a topic we'd like to introduce now because it has been ignored for a long time, yet you guys might have to use this for your treatment as you begin to practice. So it's good to know. And as you get into research also, it's an area to look at. Transduction is not the same as conjugation, but don't worry, you learn about conjugation in bacteriology. Okay. So I know there may be questions that I have missed because there are quite a number. I should have responded to this as we were moving on. Is viral DNA of any importance to the body, or it will end up causing infections? If it is phage DNA, it will not affect the human body because free DNA will be destroyed anyway, but it cannot enter human cells. It doesn't enter the human cells as such. So does lysogenic cycle mean more beneficial because of the replication of the bacteriophage genome? Well, it depends. If you're using if you're working in the lab and you want to express a certain gene, lysogenic cycle may be useful. But if you want to use the phage for treatment, you want a lytic phage, not a lysogenic one. Is UV radiation or other external stress, such as chemotherapeutic agents, considered as part of the lysogenic cycle or not? Now, lysogenic phages can be induced to go into the lytic cycle in the lab by inducing UV radiation, or in the body by chemotherapeutic agents. So any tough conditions will make a lysogenic phage get into the lytic cycle. What is lambda phage? It's just one of the phages. There are many types of phages. Lambda is just one of the phages. There are many others. Must the genome be located in the head, or it can be found in the body? It can be in the tail for phages that don't have a head. Does the specificity mean they can be used to identify and type bacteria? Yes, Kamunya, I think we answer that later. I don't understand that question. On Sanger sequencing, how exactly does the random arrangement of ddNTPs generate the order of bases of the virus or whatever DNA it is? Does the random order have the possibility of generating any random sequence? I was confused in terms for that. Well, Ramya, I think I don't know if it will be possible to explain this without an image, but Sanger sequencing will include random incorporation of ddNTPs, yes. And these ddNTPs are incorporated at different positions. So, and once incorporated, they will cause termination. So each termination will occur at a specific position. And how we identify the specific position is by passing the fragments of DNA through a gel so that they can be sorted. And the shortest will come in first. And so as they go through the gel, they'll go through a camera that will image the ddNTP. The one ddNTP that caused termination in that segment is the only one that can be imaged because it has a label. It is labeled with a dye. So for each segment, you'll only read the last DDN, the last monomer, which is the DDNTP. And as you read, you will move from the shortest to the longest, and you'll be moving this base by base. So that if you started at position 20, the next one that will go through the camera is position 21. And you only read position 21 because from position 1 to 20, the DNTPs there don't have any dye. They cannot be read. So you only read the last one for each segment. And so as the segments go through, maybe from position 20 to 200 or position 20 to 300, you'll be able to use the DDNTP that cause termination to know what is at that position. I think, I don't know if you still have challenges with this, we can arrange to do, you can read on it, and if you still have challenges, you can arrange to do a better explanation next time with the images. The questions I see here have all been answered. And I think I'll stop the questions there and go to the next slide. And this is afternoon today. The topic is antiviral agents, but I'm going to do this as an assignment because you've already covered most of the viruses and hence a lot of the antiviral agents. An antiviral agent is basically a drug that you use to treat viruses. And so please take a screenshot of this. I can just share the PowerPoint so that it's not too crowded by the other things. So take a screenshot of this, and I'm going to explain each question so that hopefully we can meet at 3:30 for 30 minutes to clarify any issues. But I would like you to start by listing all viruses. They're less than 10. All viruses for which you have specific antiviral therapy. There are not so many, maybe 5 or so, maybe slightly more, but not more than 10 for sure. And the second part of the question 1 is for each virus, you list the categories of antivirals by mechanism of action and cite 1 or 2 examples for each mechanism. So I'm going to show you an example for this and then come back to this slide. You can see for antiretroviral agents, the virus here is HIV. And for HIV, we have 5 categories of drugs by their mechanism of action, starting from attachment to fusion, to reverse transcription, to integration, and to protease inhibition. So this is what I mean by that second question. You first of all list the viruses, and for each virus, categorize the treatment or the treatment options by the mechanism of action. Once you do that, you will do the second question, which is classify antiviral agents by mechanism of action. Now this one is regardless of the virus they treat. So you classify them by mechanism of action. They are around, depending on how you break it down, 8 to 10 mechanisms that are unique. You can put viruses under, antiviral agents under these categories, and you briefly describe each mechanism. So when you say, for example, this drug, this mechanism is protease inhibitor or protease inhibition, then you can tell us that these drugs work by inhibiting the enzyme protease and thus preventing modification of viral proteins or cleavage of viral proteins. And so the viruses cannot be assembled or they cannot mature into virions. That is an example. And then you give an example of a protease like lopinavir for HIV, for example, or there are some for hepatitis C, for example. In the same question, I would like you to specifically look at acyclovir, because this is a prototype drug, and describe the mechanism of action in detail. Acyclovir is a prodrug. I would like to see you describe how it is activated from acyclovir to acyclovir triphosphate, and then how it works. So basically those elements, how it works, what is the mechanism of action, and then what viruses does it target. It is a herpes drug, anti-herpes drug, but which herpes viruses can it target? The last one, and this is barely to just demonstrate to you why it is important to understand these mechanisms, is to distinguish the mechanism of action of nucleoside reverse transcriptase inhibitors from that of non-nucleoside reverse transcriptase inhibitors. And this is in relation to DNTPs actually. Nucleoside look like DNTPs, non-nucleoside do not look like DNTPs, they work differently. So once you do that, we will convene again at 3:30 p.m. for a short discussion so that if there are any issues, we sort them out. And I will make sure at least 4 or 5 questions in this topic, if not more, come in the cut so that I make it worth the effort. And you realize that by going through this assignment, you will remember them better than if I just present them to you and without a lot of engagement on the part of your brain. So I think 1.5 hours is enough. If you finish earlier, you can do whatever else you want to do, and then we can meet at 3:30. Any questions you can ask now? Any clarifications on the assignment? If not, I see something in the chat. How do the RNA bacteriophage introduce their genome into the bacteria? Do we submit the assignment? No, I do not want you to submit it because we will not mark it. What I want you to do is, if you get stuck somewhere, make a note so that when I come back in the afternoon, you ask those questions. I'm also going to be asking you guys questions so that I find out how much you have read or understood. So really the afternoon will be a discussion rather than me presenting a PowerPoint. Okay, any questions? Yeah, we join class at 3:30. If you wish to join earlier to discuss, well and good, but I'll be joining you guys at 3:30. We will meet until 4, then you guys can start the weekend. I know it's a Friday, so I will not keep you for too long. Okay, so enjoy your lunch. We meet later in the day. Thank you very much.
Thank you.